Hey, hi everyone. My name is Ella, and my summer K fellowship project looked at understanding the soil microbiomes of riparian resources in the Alawai watershed. So before I get started, I just want to say a quick thank you to Papa Jack for allowing me this opportunity to complete my summer research. So the problem that my project was centered around is the growing water pollution and watershed degradation that we see in Hawaii. 93% of the 148 monitored marine water bodies in Hawaii do not meet water quality health standards, and the two standards that are most often not met are turbidity and nutrient content. And sources for these types of pollution can be mainly traced back to runoff pollution, which occurs when rainwater picks up harmful substances such as fertilizers, pesticides, and debris from its surrounding environment before entering stream systems, which eventually lead into the ocean. And this is especially pressing because, as you can see in the map, all the blue lines represent perennial streams on Oahu that lead into the ocean. And areas that have undergone heavy alteration for urban or agricultural purposes create this plethora of contaminants that can be carried into the stream. And high pollution at the stream level is detrimental to not only the stream ecosystem, but also its downstream marine water bodies. So a possible mitigation strategy for this type of pollution is implementing or maintaining riparian buffers, which are just a stretch of unused vegetated land around the stream that reduces erosion and filters out pollutants like sediments and nutrients before they reach the stream bank. These buffers simulate the habitat that would naturally surround the stream prior to human alteration in an effort to restore its natural ecosystem functioning. So besides the main purpose of riparian resources, which is pollution filtration and stream bank stabilization, one of its understudied advantages is its ability to cultivate and support a healthy soil microbiome. And this just refers to the diverse community of bacteria, fungi, and archaea that live in the soil that are crucial for regulating nutrient cycling, maintaining plant health, and keeping harmful pathogens at bay. So soil microbiome composition is important to plant health because out of the approximately 29 essential elements for plants, 18 are obtained from the soil, mainly via beneficial microorganisms, which are just certain microbes that carry out processes that support ecosystem functioning. For example, nitrogen fixing and phosphorus solubilizing bacteria in the soil contribute to about 80 to 90% of the nitrogen and phosphorus taken up by plants. And some other microbes in the rhizosphere specifically strengthen seed germination, seedling establishment, water uptake, stress tolerance, and hormone regulation. And strains of bacteria like Pseudomonas are also capable of solubilizing minerals and degrading pollutants like pesticides and heavy metals. So overall, healthy and diverse soil microbiomes are important for ecosystem stability, and its effects can reflect positively on water quality and watershed health. So the objectives of my project were to analyze the soil microbiomes of 8 points along the Manoa and Pololo streams, and also to identify the presence of beneficial microorganisms in areas with varying riparian zone conditions. I specifically chose to focus on the Alawai watershed because it suffers heavily from nutrient, sediment, bacterial, and chemical pollution, and it contributes significantly to nearshore water contamination. It's composed of the Palolo, Manoa, and Makiki streams, and it is the most densely populated watershed in Hawaii. So specifically the Manoa stream, 80% of its lower reaches, which you can see in the middle and the left picture, have been altered for flood control using concrete banks or fully lined concrete channels. And Pololo Stream, along with having heightened levels of all the above mentioned pollutants, has also been subject in the past to wastewater discharge, especially after heavy rainfall following storms. So my sample sites We're at 8 different places, 4 from each stream at varying elevations. The green dots are all my Manoa stream sites, and the red dots are the Pololo stream sites. And ideal riparian zones at these places would be ideally 4 times the stream bank in size, or around 35 feet, with native grass and shrubs which mitigate erosion, and ample tree cover to moderate water temperatures. So my actual sampling process just involved digging holes 10 centimeters deep on the side of the stream, 1 to 2 meters away from the stream bank. And then using the samples, I extracted the bacterial DNA from the soil using the Power Soil DNA extraction kit. And for my PCR amplification, I used the primers I used target the 16S ribosomal RNA gene, which is commonly used to identify bacteria species. And the hypervariable region in the 16S gene allows us to identify species level differences in the sequences. And then lastly, I just prepared the PCR product for nanopore sequencing using a rapid barcoding kit. And the nanopore sequencing technology just allows us to read the sequences of the amplicons in real time as they're fed through a transmembrane protein. So after we completed sequencing, we created this relative abundance plot. And the y-axis shows the relative abundance of the bacteria phylum in each sample. And the red dots on the bottom represent my Pololo samples, and the green ones represent my Manoa samples. And the notable bacteria I found in all these samples are Acetobacteriota and Actinobacteriota, which are the red and orange regions of the graph. And these play a role in biological degradation and the decomposition of organic matter. Other bacteria I found were firmicutes, which are the green region, which include strains of Bacillus, and they're known for stimulating root development and enhancing plant growth, also for disease suppression and bioremediation. And then lastly, the purple region is Protobacteria, and this includes like Cynotrophomonas and Pseudomonas, which help soil fertility and plant growth. So to better understand the composition of the bacteria in the samples, we also looked at the Simpson's Diversity Index. And as you can see here, Polo Elementary had the most diverse bacterial community. And this was one of the sites that had a relatively better maintained riparian zone. As you can see here, it had a width of around 22.5 feet on the side the sample was taken. And it also had pretty decent tree cover with different plant species like guinea grass, fiddlewood, and umbrella papyrus. Other sites that ranked higher in diversity were Wa'akawa and Manoa District Park. And Wa'akawa actually had the most undisturbed riparian zone out of all the sample sites. And all 3 of these sample sites had an elevation of around above 50 meters, while sites that ranked lower had elevations below 50 meters. Okay. And then, so my conclusions are the main beneficial microorganisms that were detected in Hawaiian stream banks in the Alawai watershed were Protobacteria and firmicutes, mainly strains of Bacillus and Pseudomonas. Also, that higher diversity was noticed in areas with higher elevations and better maintained riparian zones. And this just tells us that watershed health is not only about water quality, and soil health can act as an important indicator of overall ecosystem conditions. So future efforts to restore our watershed should address soil composition and increasing bacterial diversity. And more care needs to be taken for places that are considering channelizing their stream systems, because it is not only detrimental to water quality, but also soil microbiome health. In addition to collecting these samples and figuring out the bacterial diversity, I also took measurements on pH and soil condition. And in the future, I would look more into that aspect and how it shapes the soil microbiome, like moisture too. And then just want to thank my mentors, Mr. and he took me out to all the stream sites and just shared his knowledge of the stream systems with me. I also want to thank Mr. Hill for helping me with my DNA analysis portion and Ms. Kobayashi for always guiding me and helping me throughout this process. Okay, thank you.